Okay, so let's start Ordination 2, and we're going to begin with Lecture 10.1 on Permutational ANOVA, and we're still in the module on multivariate models. No required readings, but I wanted to include a few really classic papers by Marty Anderson and others about how to do these non-parametric multivariate analyses, so these PERMANOVAs. So we've been thinking a lot about multivariate analyses and considering that there are several ways to look at multivariate patterns from a matrix of Y's. And so we're going to be talking about permutation tests, or PERMANOVAs, to actually test for patterns in community data. We'll be revisiting our zooplankton data for this PERMANOVA example. And so again, if you recall, we were thinking about the question, how does increasing connectivity of waterways alter aquatic zooplankton communities? And this is from a paper by Strucker and Britton in Journal of Applied Ecology in 2017. And so when we were talking about nMDS, we created a figure of these zooplankton communities. So each point represents a sampled water way of some kind, some sort of water sample, and in each sample the abundance of different zooplankton species was calculated and measured. And then each of those water samples, each of those samples had a bunch of different variables. So they had, and those are represented by these arrows, right? So Secchi depth or elevation of the site sampled, etc. And so that's what this figure represents. But this doesn't tell us any statistical relationship between these variables and the communities they are plotted on top of. But before we get into figuring out how to do PERMANOVA, I wanted to bring up a few things that people had questions on in class last time. So there's a fantastic function called dimchecks MDS to look at the dimensions. So I gave you code to do this by hand, but there's a really nice function in Go eveg that allows you to look at any number up to any number of dimensions that you want, and with your distance matrix and your data, and you can look at what stress each dimension is producing. And it looks like here their line is at 20% stress as sort of their mark of if the stress is low enough to be understandable and acceptable. And so my criteria of 10% last time might have been a little strict. And I also wanted to show you all how to plot these nMDS plots in 3 dimensions. So if you end up using 3 nMDS axes, you can do that. So you use the vegan 3D library and you also have to have the RGL library installed. And when you do this, not in Markdown for PowerPoint or for PDF, these figures are actually interactive, so you can rotate them. So this is the code you use to create these figures. But I just took a screenshot and put it on here so you could get an idea of what it looks like. Okay, so let's talk about relationship between your X variables now and the site patterns in community level data. So how do you determine if there's a relationship between the plotted environmental variables and the site level community patterns? So you could try principal components analysis or principal coordinates analysis, and with these methods you would use dimension reduction and then use some of those dimensions back into looking at more univariate models potentially. If you want to look at the effects of the community pattern, you can also use canonical correspondence analysis or redundancy analyses. And these are fairly common in the literature, they're fairly common in the literature, but we're not going to talk about them here. But I will note that for both, relationships and predictors are forced to be linear. And they're typically non-distance based. But you could try db-RDA. That's a newer method. But finally, there's permutational ANOVA or PERMANOVA. And this allows you to determine if the relative lengths of the arrows that represent environmental variables in your nMDS plots are predictive. And so it actually allows you to run a statistical test to determine if certain variables predict your community outcome. Permutational ANOVA, or a PERMANOVA, allows you to make to basically run statistics on if certain environmental or certain X variables are significantly predicting a matrix of Y variables, right? A community score. And so given a set of site scores, axis 1 and 2, etc., and environmental information at each site, the PERMANOVA procedure calculates an R-squared to determine if site is a function of environment for each iteration. And so the null hypothesis is that there's no correlation, that the correlation equals 0. And so what PERMANOVA does is it holds the Y data constant and shuffles the X's, or your environmental data or treatment labels. And so say your actual data looks like the picture on the left with a strong correlation, where your environment 1 predictor is sort of positively pointing towards the top left corner. What would happen is that you would run a permutation where you hold your community data or your Y matrix constant, and you shuffle your group labels or all your environmental data, and then it would rerun the nMDS to find out if, what the correlation is between all of your communities and your site level variables, and it would calculate an R-squared. And so when the PERMANOVA ran this certain permutation for this no correlation example, you can see that now all of the sites are shuffled up in space, and there's no environmental variable that is now coming through or is correlating with the new locations of each plot. And so what PERMANOVAs do is that it runs permutations thousands of times, and the relative location of your observed R-squared from your actual data in relation to the permuted R-squares indicates association strength. So if you simulate your relationships 1,000 times, if your observed R-squared from your actual data falls in where that first red line is, about an R-squared of maybe 0.2, then if it fell there in the distribution of permutations, then there's no evidence of an association, because by random, you're getting a lot of permutations and options for your system that have a stronger correlation between the communities and the environment than the particular one that you observed. However, if your observed R-squared falls in the tail, sort of right around 0.75 in this picture here, there's really strong evidence of an association. So if you shuffle your X values in your environment in association with your communities, your zooplankton communities 1,000 times, and your observed data still has a stronger correlation between the environment and the communities than all of your permuted, your 1,000 permuted communities, then you have a lot of evidence, right? If it falls in that 95% tail, 97.5% tail, you can say of that distribution, you can say that, that's equivalent to a p-value, a significant p-value. So how do we do this? This actually all happens under the hood. You can do it by hand, but you just can do it with the adonis function if you'd prefer. So let's examine how community pattern is a function of Secchi depth and site type. So if you remember, Secchi depth is how clear the water is, how deep how long it takes for basically how deep a disk is when you can't see it anymore. And your site type, and that was like lake, canal, river, etc. And so FYI, this analysis deals with X's sequentially, so the order of your terms really matters unless your data is balanced. So to run this, we have to create a distance matrix of our zooplankton, and remembering to drop that first column of the data because that is our site that's our plot numbers. And we want to use a Bray-Curtis distance here. And so we're going to create our Bray-Curtis distance matrix, and then we're going to run that with an adonis as a function of our two x variables, Secchi depth, and type. And again, specify our data. And here we see that both Secchi depth and type of lake have significant effects on the community of zooplankton. And you may want to add a blocking factor. So you can include a block effect in your adonis with your strata equals. And so here permutations will happen within these groups or blocks. So permutations will happen, for example, within lakes and canals and all of the different types of, because we're going to stratify by type here. So to do that, you again run your adonis function with your distance matrix as a function of, now we're just going to look at Secchi depth, specify our data, and here we make sure to specify data as the environmental data, because that's where we're getting our x's from, right? Same as before, sorry. And the strata is the type. And so when you run that, you can see that within these different types, Secchi depth is still significant. And this is sort of, blocking in this way is sort of equivalent to a random effect, sort of. So you can just sort of think about it that way, like taking into account the different types of water bodies sampled for zooplankton communities. Secchi depth is still significantly a significant predictor of those zooplankton communities. And so I did my best to create some prettier nMDS figures because they were called for. And so you can see the code under the hood that allows you to change the color. I don't think we didn't do ellipses last time that go around the data points, and so I wanted to draw those here. Also, you can color your spider diagrams, and I also have the code under the hood for that, which I think these look really nice. One really cool thing that you can do in this, when thinking about adonis and in this package, is you can look at the dispersion within your groups with the beta disper function. And so if I wanted to look at the sort of how, it's one way to think about beta diversity or turnover among plots, but that you can also think about it as how large are the circles. So in this figure, both pond and lake have very large dispersion, right? There's a lot of variability, or within plots of a certain type, but reservoirs are much smaller. And you can actually see that from the spider graph, where you can see that those lines to the centroid are much longer for lake and pond than they are for what reservoir. And so that means that lake and pond have more dispersion than the reservoirs do in their community structure. And so you can actually measure that dispersion with the beta disper function. And so if you use your distance matrix and then look at whatever environmental variable you want to look at the dispersion within. This works best for categorical variables. You can see the average distance to the median point. And so here again, you can see that ponds and lakes have fairly large dispersion, which is about 0.56, 0.57, whereas the reservoir had 0.33. So again, smaller dispersion. And so a few things I wanted to point out is that you can extract nMDS scores for the number of dimensions that you chose, excuse me. And so if you want to, like if you did an nMDS with 2 dimensions, you can pull out those nMDS scores with points, asking for the points, and this allows you to create your own figure. If you wanted to just point those as X and Y points, you totally could. You get one for each of your samples or communities here. And you can also use it for the dimension reduction that we're going to talk about sort of as we move forward. And you can do this for as many dimensions as you specify in your MDS model. So here, if you do one with dimension of 3, you can pull out your 3 nMDS scores, etc. And I wanted to, again, I found another really nice way to plot. The website for this plotting is in the code under the hood for this, but this is sort of those surface plots, but an ordy surf for a nMDS plot, but you can do it in ggplot in a way. Again, all the code's under the hood and make it look really, really nice. And so I thought this was a really cool looking way to show the surfaces by using colors of the lines instead of having numbers, which can be really difficult to read at times. And so I want to leave you with a few nMDS and PERMANOVA notes. So one thing that was asked in class was, is it important to set a seed? If you want your results to be reproducible, or mostly reproducible, I should say, it's not going to be 100% reproducible. That's a bit of an exaggeration. Make sure you can set your seed before you run your nMDS, because the nMDS is an optimization process. There, over the multiple optimizations, there might be a slight difference in your outputs at the end, but so that's why it's not 100% reproducible, but you'll get a lot closer if you set your seed to getting the same thing every time. And you also want to make sure that your results are not dependent on that seed that you chose. So try a few just in case. And then PERMANOVA does a lot better with larger sample size because it has more to work from when it's randomizing. And also PERMANOVA works best with balanced data that's homoscedastic. So I wanted to include a metabolomics example, but it was going to take me a bit longer to get the slides together. And I wanted you all to have more time this week to be able to look through your notes and your lectures. So I'm going to say that I will put this up towards the end of the week so you can see another example with a different type of data set on how to do PERMANOVA with metabolomics data. But for now, I want you to be able to get through the lectures. And with that, please complete quiz 10.1 on Canvas and continue on to the next lecture.